So it's hard to believe we only have a couple weeks left. Alright, so for the final exam, as with the midterm, right, all your homeworks, your quizzes are excellent study guides, right, so make sure you have all of them. You should always know your terms that were assigned in the homework. Remember, you do need to be able to spell terminology. The PowerPoint slides, especially the scientific method ones, are excellent ones because they summarize each experiment. Exam covers all the exercises after midterm. Most of the experiments helped you identify your unknown, right? But some are standalone exercises that don't relate to the discovery of your unknown. So, wet mount and hang drop, right? Which, did I end up doing a hang drop to you guys? Do you guys see a hang drop? Okay. We'll try to set one of those up next week for you guys. But you guys saw the videos, we talked about that technique and why you would utilize it. You do need to know that. You did the motility test on your unknown, right? So you need to know what that test tells you, right? All the components, how it works. Same thing with fluoroglycolate media. Why do we use it? What does it tell us? All the components of it. Phenol red. You need to understand how the methyl red test works and the Voges-Proust-Calver, right? All those things. Citrate, catalase, mannitol salt, Columbia CNA, and the eosin methylene blue, right? Everything we've done on and related to your unknowns, you need to know in the detail that we've covered it, right? So everything possible. So homework, quizzes, PowerPoints, excellent source of review and study. Standalones. I know you guys have probably forgot about this, but remember standard plate count and dilution calculations? Yes, that is on the exam, right? So review that. If you're still struggling with that, make sure you come see me, right, and get help on that. I've lots of videos and remember lots of worksheets for you guys that were online as well. But definitely if you're struggling, come see me because you will have a dilution problem on the final exam. Antibiotic sensitivity, the Kirby-Bauer, remember those discs we measured? Again, I'll give you that chart. You just have to be able to interpret that information and understand how that test works. Remember, anaerobic jar, we talked about it, but we didn't actually do it. But again, you still need to know all the aspects as it relates to that experiment. Bacterial transformation is what we're going to do next week using the P-Glow system. We're going to make green glowing E. coli. Pretty cool. And then the last lab, you're going to do ELISA, and the type of ELISA we're going to utilize to be able to detect the presence of antibodies. And that's referred to as an indirect ELISA. There's two different types of ELISAs that we'll talk about. So you should understand the process of identifying an unknown organism as well. All the steps you performed to identify your unknown organism. Evaluate stain slides, right? So again, I'll have microscopes set up and you've got to tell me whether the result is gram-positive or gram-negative. You've got to tell me which one has a capsule, which one has a spore, which one's acid-fast, right? Be able to classify morphology still, right? What's the shape? Is it a rod? Is it a cocci? Is it in clusters? Is it staphylococci? Is it forming chains? Right? Is it streptococci or streptobacillus? You still need to know those. That's kind of a carryover from the beginning of the semester. Given a table of known data, right, you got to figure out what particular unknown that person may have had, right? And so in this packet, we've got a couple of examples that we'll go through so for some more practice, because you've only done this once, right, on your unknown. So again, you don't have to memorize this table of data. I will give it to you, right, for the exam. You're not memorizing, right? You're taking in information and evaluating it based on known information. Does that make sense to you guys? Okay. So this is the first question, right? So a student has obtained the following results on his or her own unknown. It's gram-positive. The phenol red broth test was K. So what color was that tube? It was pink. Motility test was negative, which means it just looked like a straight line, right? There was no diffusion into the media, no motility. The capsule stain was negative, and the acid fast was positive. Which organism is this out of the list of organisms? Mycobacterium. What made you get that so quickly? The only one that's acid fast positive is Mycobacterium, right? So right away, right, if you remember and recognize that, especially if you look at this chart, right, acid fast positive is only one. Only Mycobacterium, right, are acid fast positive. But then the other tests would help confirm that. So for the next one, gram positive, phenol red, acid, no gas. So what color is that tube? Yellow. Is there a bubble? No, because no gas. Motility is negative, capsule stain's positive, catalase is positive, and citrate is negative. What you getting? So look at the table, right? Look at that data, see what you can figure it out or narrow it down to. Two of them, right? Not enterococcus, gram positive. The two staphs. Staph aureus and staph epi, right? Have those same results for all those tests. So let's look at this, right? Gram, that divides our thing in half, right? We can cross out all the gram negatives. It leaves us just gram positives. When you look at phenol red, it eliminates 2 of the choices because they were K. So that leaves us with 6 choices. When you look at the citrate test, it's negative, so that eliminates all the bacillus, which are all positive for citrate. When you look at catalase, it eliminates enterococcus faecalis because it's negative for catalase, so that leaves you with the 2 staph. So, when you look at these guys, the only difference is what? The VP test. So, you're going to rely on just one result? No. You want additional results. So what plate media would help us differentiate Staph aureus from Staph epi? MSA, because we know that Staph aureus is going to be what? Yellow, because it ferments the mannitol. And another thing that I don't expect, this I do expect you to know for final exam, right? You should know that Staph aureus is yellow on these plates. What's another plate media, though, that would have been beneficial? The CNA. So if you guys notice in your unknown report, on the CNA page, it lists what organisms are alpha and beta. You'll notice that Staph aureus, not Staph epi, is listed as beta hemolytic. You guys do not have to memorize beta or alpha hemolytic organisms for the exam. Okay. This is the last one, right? Oh, we have 4 of them? Okay. So, this one I think you can narrow it down to a single organism, right? Okay. See what you get. Do you guys want me to read results, or is that screwing you up? You can pull those 2 pages apart, you know. I just have them together for handing out purposes. Think we got it? Let's see if you guys are right. Serratia mercescens. Did I go over pronunciation guide with you guys last time? No, okay, that's one thing I want to point out to you guys. Did everybody get that one? Okay. Alright, so gram negative, so that eliminates all the gram positives. Methyl red eliminates 2 of the negative results. Citrate narrows it down to the 2 positives, which narrows it down to Serratia and Klebsiella pneumoniae. And then the phenol red helps narrow it down to Serratia mercescens. Okay, last one. I think this is another one where you got to narrow it down to 2, right? Spore positive. That brings it down to 3, right? It's got to be a bacillus. Remember, negative for anaerobic growth meant that it only grew at the top of the FTM tube. It was aerobic. So which 2 bacilli? Yeah, Megatherium or subtilis. So again, spore brought us right down to 3 possibilities. Phenol red and anaerobic growth narrowed it down to the 2. Now, one test from the chart. We're not talking about media, but what test from the chart would help us differentiate these 2? Right, the Vogts-Press-Kauer. Because notice that's where they differ, right? Just that one test. Again, would you rely on just that one test? No, something could go wrong, right? So, again, for these 2, if you had them, what would help confirm is one of the plate media. And in this case, what is it? Amy? Which plate media helped you confirm that you had Bacillus subtilis? Yeah. Was it the CNA, the EMB, or the mannitol salt? Nope. It was the CNA. Because if you notice on the CNA list, yours is one of the betas, right? And I showed you another culture that I had done that came out, and you could see as beta. Again, you guys don't have to memorize who's beta and who's alpha, right? But understand that one of the reasons why we did these tests is to help confirm your results, because in some cases we were getting down to just one result, and if that was off, right, you may misinterpret as to what your unknown is. And then I did show you guys last time that NRO tube, that tube where they would test a whole bunch of different tests all in one fell swoop. They wouldn't do the individual tests necessarily like we have in a clinical lab. So the first half of our course was very fundamental, right? You basically learned how to stain, right? And then now we applied that, right? We looked a lot at structure. So we still looked at that here. But then we also looked a lot at functionality. What can these organisms do, right? We talked a lot about fermentation, right? And hopefully, you know, for those of you guys that maybe didn't cover it so well in biology class, right, you have a better understanding now of glycolysis and Krebs cycle and the importance of these things in fermentation as well. So what our last 2 labs are going to focus in on is genetics, right? We're going to actually genetically modify bacteria next week. And then we're going to also look at serological, how antibodies can be used in detection of pathogens. So remember, for our microscopes, the reason why we're using them is they increase resolution, provide magnification, and contrast. is generated by staining. So again, you got to know your results, right, for each of the staining procedures, right? So I'm not going to give this PowerPoint to you guys, but this is a good example, right? When you're studying, maybe generate yourself a table, right, of what the results should look like and what they tell you. It's more detail than I want to go into. So standard plate count, as I said, remember it's important to know the aspects that plates are only countable if they're between 30 and 300 colonies. The formula that you need to know for determining the original concentration per milliliter, right, this formula. So remember, you need to know the dilution factor of the tube in the series. You need to know the volume that's plated, and you need to know the number of colonies, right? And you can only count ones that are between 30 and 300. Some of these slides I might actually post for you guys. I don't necessarily have time to go over this. This is a good example, right? Antibiotic sensitivity. Right, make sure you go over that. Remember problems that we can run into with controls, right? If we have a false negative, it might be a sensitivity issue of the test, right? Not enough of the product to be able to visualize that is actually a positive result. False positives is usually a specificity issue, right? Something other than what you're testing for is causing that positive result. Remember, we can run into that problem with the catalase test if it's not performed properly, if you use a metal loop, because it can cause the bubbling instead of your organism. Most of the time, with most of the tests, right, it was a sensitivity issue for us. But sometimes specificity can be an issue. Determining motility. And we just talked again about fermentation today. So I might go over some of this if we have time next time to help review. But I am going to post this one for you guys. I don't know how many more slides is in this. Oh, just a couple more. I'll post it later, but I'm not going to post it now.